So far into genetic technology, we find that this is a very interesting chapter because it's about applying the basic knowledge to design processes or procedure as well as to produce products that are useful for human. So in today's lesson, we will further continue to explore another process that is being designed in genetic technology that is also very useful for us. So the learning outcome says that candidate needs to be able to describe and explain how gel electrophoresis is used to separate DNA fragments of different lengths. So first of all, we need to know the definition of gel electrophoresis, right? So it is a separation of charged molecules, which is example DNA, by differential movement. Differential movement means different movement rate through a gel in a liquid medium in an electric field, and the degree of movement is dependent on the mass. Mass means either size or length of the DNA fragments. So this is a full definition. So I'm going to explain the detail now. Okay, so you can see on the right side, this square, this rectangle is actually the gel, and you can see bands moving. So each of the band is actually a DNA fragment of different sizes. So we make the DNA start to move in an electric field. So you can see the positive negative sign here denote electric field across the gel. So when there's electric field, DNA will start to move. And then when they start to move, they start to separate based on their different sizes or length. Now, the gel appears solid to our eye. The gel is like, you know, the agar that you eat, right? But this gel cannot eat one. You cannot eat. So the gel looks solid, but actually it's colloid. Now, colloid in this case here means solid in liquid. So if you look at this magnified version of the gel, so if you magnified it, okay, we will see that actually it's made up of solid with a lot of pores in it. So they are actually the gel fibers with a lot of pores in it, so the spaces in it. So these spaces between the gel molecules, so those fibers are the gel molecules, they allow other molecules to pass through it. So they act as a barrier, okay, so that larger molecules are difficult to pass through. Smaller molecules can easily pass through the pores in the gel. So that's how we separate the DNA in the gel. So why can we separate DNA in the gel? It's because DNA, you know that the structure of DNA is made up of sugar, phosphate, and nitrogenous base. So therefore, we have got the phosphate, sugar, phosphate, sugar, phosphate, sugar in the DNA. And then the bases are joined to the sugar, right? So this is how they are arranged in the polynucleotide strand. So these are the nitrogenous bases. And so if you look at the one polynucleotide strand of the DNA, you see that there's this region that will be charged. So can you tell me exactly which region is being charged? Asmira, look at the diagram and tell me which part of the DNA is charged. Phosphate. Yes, very good. Phosphate. You see, for the phosphate, the O that has not formed any covalent bond with another atom, it will be charged. So it's negatively charged. So every phosphate in the sugar phosphate backbone is negatively charged. So the entire DNA molecule is highly negatively charged because all the phosphates are negatively charged. So because DNA are negatively charged, we can actually separate them based on because they have the charge. Okay, so therefore the electrodes are placed at either end of the gel to create the electric field, and then DNA that's negatively charged will then, due to the phosphate group, will then start to move towards the positive electrode. So we place so if this is the gel, so if you place the DNA over here, so let's say we put the DNA over here, okay, so this is a piece of DNA, then we know that because DNA is negatively charged, so what is the electrode I should put near DNA to make it move away? Annabelle, so what charge should I put here so that the DNA will start to move downwards? Annabelle, what charge should we use? Positive? Yes. No. You want it to move down. You want it to move down through the entire gel so that the DNA with different sizes will start to separate. So we must use negative charge, right? Okay. So we must use negative charge. So your negative electrode will be placed here and positive electrode will be placed here because the DNA are negatively charged. So it will start to move away from this negative electrode towards positive electrode. Where's my positive? Positive electrode. So this is positive electrode, positive. Okay, so here. Okay, so it will move towards the positive electrode here. So start to move this way. So that DNA fragments of different sizes will start to separate into different location. So the rate of movement of the DNA depends on its mass. So the smaller DNA will move faster than the longer DNA. Because the smaller is the DNA, it's easier for them to pass through the pores within the gel. So let's look at the factors which will affect the movement of the molecules in the gel during electrophoresis. So firstly, the movement depends on net overall charge of the molecule. So because you place the molecule at the negative electrode, so if the molecule is negatively charged, it will be repelled and start to move towards positive electrode. So if it's positively charged, it will remain here. So let's look at this one. So a molecule that's negatively charged, anion, will move towards anode, which is positive charge. A positively charged molecule, which is cation, will move towards cathode. So remember, anion moves towards anode, cation moves towards cathode. If the molecule is highly charged, it will move faster because attraction between positive and negative becomes stronger. So second factor affecting the movement of molecule is the size. Size for DNA means the length of the DNA or also the mass of the DNA. So the distance traveled by the DNA molecule is inversely proportional to its length. That means the longer is the length. So the longer is the length, the shorter is the distance traveled. Because the heavier it is, the longer it is, the harder it will move, or the slower it will move. So the distance traveled becomes less. So smaller molecules will move faster than the bigger molecules. So thirdly, pore size of the gel. Smaller pore size will allow smaller molecule to move faster through it than the bigger molecules. So these are the three factors which govern the movement of the molecules. Number one, net overall charge. Number two, the size of the molecule that you want to separate. Number three, the pore size of the gel. So this is to look at the procedure to set up the apparatus to run gel electrophoresis. So firstly, we need to prepare the gel. Preparing the gel is like preparing, if you have done make agar before at home, it's very similar. So you take the powder for the gel, add it with water, boil it, okay, and then cool it down to 65 degrees Celsius. So after that, you pour it into a mold. So this is a mold for us to make the gel. And then this blue color structure is called the comb, C-O-M-B, comb, because it looks like our comb. So there'll be a comb that's placed in the mold to create. Can you see that it creates this little tiny groove inwards towards the gel? So all these tiny little grooves are called the wells. So the comb is used to make the well. So to make the wells. So allow it to set at room temperature. After it sets, we can transfer the gel into the electrophoresis tank. So this is the tank for electrophoresis, because you can see that you got this left side and right side is the electrode for you to join the electric wire to it. Okay. So you can see here in the gel, now there is this well, right? There's a well. So the next step is we will actually use micro pipette to transfer tiny solution of DNA fragment mixture. DNA fragment mixture means that this sample has got DNA of different sizes, because how the DNA is being obtained is that it's from the chromosome of individual, and then we use restriction enzyme to cut it. Okay, so the restriction enzyme will cut at the restriction site. So it depends. If this piece of DNA does not have the restriction site, it will not cut it, right? If this piece got restriction site here, 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 it will cut it. So therefore, after you cut the DNA from the nucleus of a person, then you get many fragments of different sizes of the DNA. So therefore, then you take this entire sample, which is in the liquid, and then transfer it with micropipette into the well. Okay, and then inside each well, there will be a number of DNA fragments of different sizes from an individual or from a sample that they've obtained. Okay, so and then because DNA is colorless, so they will also add stain to the well, so that it will stain the DNA, so that when the DNA start to move, we can see it moves like a blue band. So we can see where the DNA has traveled. Okay. So here you can see that, so now the well is ready already. So the step number 2 is we use micropipette to transfer a sample of the DNA fragments into each well. And then we also add the tracing dye. The tracing dye is for us to trace where our DNA have traveled when we start the electrophoresis process. So now you see here, you've got negative electrode and positive electrode. That means we already started the electric field. So the DNA fragments will start to move towards the positive electrode. So it will move based on its size. Okay, so the smaller one will move the fastest, the biggest one will move the slowest. So because of the tracing dye, so we can see where the bands have moved, so we know when to stop the electrophoresis. So you see this band has, the first band that moved the fastest already almost reached the end of the gel, so we have to stop the electrophoresis here. So this animation here actually shows in real life, when you're running gel electrophoresis, what you will see, right? You start to see the DNA fragments will start to separate, and then bands at different locations will be formed. So each band contains DNA of a certain length. Okay, now can you see the first well here? Usually in the first well, we will place standard DNA fragments. Standard, remember what is standard? Like standard glucose means you know its concentration. You prepare it so that you know its concentration, right? Standard DNA fragments means you have prepared these DNA fragments so that you know their fragment sizes. So it's a reference point for you. So these standard DNA fragments are reference DNA for you to refer to it to roughly know what is the length of the sample what is the length of the sample? What is the length of the DNA in this band that has separated from the sample? So let's say, for example, so these are the bands, right? This is the band here. So if I want to know what is the length of the DNA in this band, I can always refer back to the standard, because in the standard, I know the size of this one. Let's say this one is, the length is A base pairs. Okay, this is B base pairs. So this is C base pair. So therefore, the length of this DNA is between A and B base pairs. So therefore, standard DNA fragments are actually used as a reference DNA for us to know what is the length of the DNA based on the bands, that the location of the band, based on the location of the band. Okay. Okay, so this is an actual instrument that has usually being used in the lab. Even in our university, they also use exactly the same apparatus to run the gel electrophoresis. In the degree program, they use this one. So if you've got opportunity, you may want to join their workshop so that you can see how this is being done. So what you can see here is you've got a gel right at the center with the well, and then the DNA sample have been pipetted into the well, and then the tracing dye, which is blue color, usually is methylene blue, is also being added in and mixed with the DNA. Then you've got the electrode, the positive and negative electrode, and this is the carbon fiber which conduct the electric, okay? And then the entire gel is immersed in buffer solution so that it will conduct electricity. It's not in the space. In the space, you cannot conduct electricity, right? Because this electrode is stand alone and then separated by space to here, so it will not conduct. So you must immerse the whole thing in an electrolyte solution, which is buffer solution. So this is another diagram to show the side view of the apparatus. So right in the center is the gel. So the well is here where you pipette the DNA to the well, and then we have electrode on the left and right side. So you've got the negative electrode and positive electrode, and the entire gel is immersed in the solution called buffer solution or electrolyte. So this electrode is actually made up of carbon fiber blocks. Okay. Carbon fiber blocks is entire block here. It's entire block here. Okay. So this procedure, I will not go through with you already. I already explained the procedure. The only thing I want to mention is the procedure, how to set up and run gel electrophoresis. I've already mentioned. So you can read this as your notes. The only thing I want to bring to attention is this part here. Now, for the gel that's being used, there are two types. Yeah, either agarose gel or polyacrylamide gel. So agarose gel is being used to separate fragments where the DNA fragment sizes are between 100 to 50,000 base pairs. Whereas polyacrylamide gel is used to separate fragments where by a difference of one nucleotide only. So can you tell me between these two gel, right? Which gel has got a smaller pore size? Akila. Which gel do you think have got smaller pore size? Akila? So the answer is polyacrylamide will have got smaller pore size, because when you use polyacrylamide gel, you use it to separate DNA fragments with a difference in length of only one nucleotide, you know. This is my short form for nucleotide. The difference in length is one nucleotide. That means your pore size has to be very small so that a slight difference in the length will affect the movement of the DNA. So polyacrylamide gel has got smaller pore size. So the rest you can read on your own. These are all the information I have gone through with you already. Okay. So the next step. Okay. So once the DNA has been separated, okay, once you finish gel electrophoresis, the next step, the last step is we have to locate where all the DNA fragments have separated. The reason is because the methylene blue will not stay very long. in the gel. It will start to diffuse across the entire gel and the band is no longer visible. So therefore, we need to make all the bands visible after the gel electrophoresis. So we need to locate the bands of the separated DNA fragments. So the bands here means the separated DNA fragments. So since DNA fragments are invisible, we need to make it visible. So there are two ways that scientists will make them visible. Number one, either they use staining, or number two, they use DNA probe. So when they use stain, they will choose a stain that can stain all the DNA fragments, so all the DNA fragments will appear as bands in the gel. Then you can take a photograph of those bands and keep it in soft copy, because the gel is very fragile. So, and then you cannot keep it for very long because it'll dry off. So eventually they have to take photograph and keep the photograph for future reference. Okay, so the stain that has been used so far are ethidium bromide, methylene blue, and now blue A. So for ethidium bromide, right, they will immerse the gel into ethidium bromide solution for some time for it to stain the DNA. After that, they will wash away the ethidium bromide solution, and then they will take the gel and then they shine UV light into the gel, onto the gel, and then the bands will fluoresce because of ethidium bromide. And then they'll take a photograph of the gel. So, but ethidium bromide is very toxic because it's carcinogenic. It has been used widely in the lab, but it's very carcinogenic. So it's be very careful when we handle the solution. So second way to make the DNA visible is we use DNA probe. Now, you know, staining stain all DNA fragments, right? But when we use DNA probe, we only, because it is DNA probe is a short single length of DNA, it can only bind to specific DNA fragments containing a specific base sequence. So let's look at how the DNA probe works. So see, let's say, for example, this is the DNA that we want to find out in the gel, its location. So this yellow color band is actually a short length of DNA with bases that is complementary to the bases of this DNA that we want to locate in the gel. So this yellow color band is called the DNA probe. Okay, so what is DNA probe? So it is a short length of single-stranded DNA, usually about 100 to 1000 bases long, with base sequence that is complementary to the DNA that we wish to find or we wish to identify from the gel. So where will the DNA probe bind to? So we can use it to bind to a specific gene or allele, which contains specific base sequence. For example, let's say, for example, in genetic engineering, remember, one of the ways to obtain the wanted gene is from DNA to DNA. That means you go to the nucleus and you isolate all the chromosomes from the nucleus of the cell, and then you cut all the DNA using restriction enzyme into fragments of different sizes, and then you run gel electrophoresis. So all the DNA fragments will separate, right? So let's say, for example, this is your piece of DNA, right? And then your gene for, the gene for insulin is located here, say for example. So then you cut this DNA and of course other DNAs, because in the nucleus you've got many DNA, long and short, right? So then you cut all the DNA with certain restriction enzyme. So you cut the DNA with restriction enzyme. Okay, so you got many fragments. So once you got many fragments, this is your sample from the person. You run gel electrophoresis. So those DNA fragments will separate into different bands based on their length. So only one of the band will contain our insulin gene, right, which is this one here. So in this case, we only want to identify which particular band contain the insulin gene. So because of that, we don't want to use the stain, because the stain will stain all the DNA fragments that have been separated. So therefore, in this case, we'll use DNA probe. So we use the DNA probe with base sequences that is complementary to the sum of the base sequences in our insulin gene. So it will only bind to the band containing the insulin gene. So this is how we fish out this particular gene from the rest of the DNA in the nucleus. So the gene probe will only bind to no more than one fragment. Now, so DNA probe can bind to the gene. DNA probe can also be designed to bind to VNTR region. VNTR stands for variable number tandem repeats. What is this? I will elaborate more later on. So now you know there are two types of DNA probe. Either it can bind to the gene or it can bind to VNTR region in the DNA. So when we use DNA probe that binds to VNTR region, this is used in genetic profiling or genetic fingerprinting process, which I'll elaborate later on. Now, VNTR is a common repeated base sequence that is present in most of the DNA fragments. So therefore, it will actually, the DNA probe will bind to most of the DNA fragment to give the genetic fingerprint of the person. So different people will have got different genetic fingerprint. Later on, you'll see the reason why, okay? So the DNA probe that we used just now, right, is also DNA. So it's also invisible, it's also colorless. So we also need to make the probe visible by labeling the probe. So when you say labeling the probe means we will either add radioactive labeling to the probe. So that means we add, let's say, phosphorus isotope to attach to the DNA probe. So wherever it binds, that region will emit radioactive radiation, which can actually expose the X-ray film, forming banding patterns on X-ray film. So whenever you use X-ray film, it's called autoradiography. Okay, or we can use fluorescent stain to make the probe visible. So we can stain the probe with fluorescent stain to make it visible. So we can use vital red to stain the probe, and then when we shine UV light onto it, it will actually fluoresce. So it will allow us to know the location of the probe, and then the location of DNA fragment that we want to locate will become visible. So let's look at the diagram here. So this is the result of gel electrophoresis. So then the radioactively labeled DNA probe is actually placed into the gel. So this radioactive probe, we put in a gel by, the probe will exist in the form of solution. So in the solution, we've got this DNA probe. So we will immerse the entire gel into this solution and then mix the solution gently to allow the probe to bind to the relevant DNA fragment. So after it binds to relevant DNA fragment here, because it's complementary base pairing with it, and then we will do autoradiography. That means we'll put an X-ray film onto it so that those radioactivity will actually expose the X-ray film. Then after that, the X-ray film will only show these 3 bands and nothing else. So this is how we use the DNA probe to locate certain DNA fragment. For example, the insulin gene. Okay. So this can be used to locate. Maybe we want to know whether this person is homozygous dominant or heterozygous dominant for eye color. So we can use it to locate whether it contains the recessive allele or not. So we can use this probe to locate the recessive allele, that means blue eye. So if there's no band, that means this person is homozygous dominant, brown eye. Okay, for example. So now we want to look at genetic fingerprinting, right? So we said, okay, every one of us got our own fingerprint. No, not every one of us also got our own DNA profiling or genetic fingerprinting. That means our DNA are all different. So this process is being designed by this scientist, Alex Jeffrey, in University of Leicester in 1985. So he designed this process that every one of us got different DNA, so we can do a DNA profiling as an identity of a person, to identify the person. So genetic fingerprinting is also called DNA profiling. That means find out the DNA of a person, or genetic profiling. So this is an analysis of individual's DNA in order to identify the individual from which the DNA was taken. So we got a sample of DNA and we want to know that this DNA comes from which person. So we can use genetic fingerprinting to find out. Okay, especially when you go to a crime scene, right? You do not know who committed the crime. So you try to collect samples like blood samples, semen samples, what else? Hair, you know, the hair, as long as your hair contains the follicle, the cells, right? Then the cells got DNA, so we can actually get the DNA and we can do genetic fingerprinting. So then we can trace it with the samples from the suspect and see which of the suspect was actually present in the crime scene. So this is used to identify the individual from which the DNA sample was taken, and also is used to establish the genetic relatedness of individual. That means, for example, this is a child. We want to know that whether who is the father of the child, right? So therefore, we can actually find out using genetic fingerprinting, because a child's genetic fingerprint, right, will be similar to both the mother and father. So we can use this method to find out whether who is the father. So every individual except identical twins will have unique DNA profile, because they are all genetically variable. So therefore, genetic fingerprinting has been used in identity matching in forensic science to find out who was in the crime scene, who committed the crime. So the high probability of who is the person who committed the crime. So to identify someone from a blood sample, root of the hair, saliva, semen, where these samples are obtained from the crime scene. And all these samples must contain nucleus, then only we can obtain DNA samples to run gel electrophoresis. Or for inheritance matching. So we want to trace paternity, means who is the father. We want to trace lineage, means how closely related are these individuals. So to identify the father of a child, and also for animals, to determine whether individuals of endangered species in captivity, that means we capture the animal and then we put it in the zoo. Okay, and then they are categorized as endangered species, like panda, for example. So we want to know whether the individual of endangered species in captivity have been bred or captured from the wild. So that means we will know, depending on how related are their blood samples, how related are their genetic fingerprint with those organisms from the wild versus those that are bred in the zoo. So this is an example of the genetic fingerprint. So we have got a specimen from the crime scene, and we've got these 4 suspects where we obtain their blood sample and we run gel electrophoresis to get their genetic fingerprint. So based on this data, can you tell me which suspect was present in the crime scene? Ki Jingyi? Which suspect was present in the crime scene? Jingyi?
Suspect 3.
Suspect 3, correct or not? So let's see, suspect 3, yes, because you can see that the DNA, the genetic fingerprint of suspect number 3 is exactly identical to the genetic fingerprint of the sample obtained from the crime scene. So therefore, can you see this is such a powerful tool? It's a very powerful tool for us to identify who this DNA sample belongs to, especially in forensic science. So once the DNA fragments are separated by gel electrophoresis, different people will give different distribution pattern or bending pattern. So this is actually called the bending pattern, right? Or distribution pattern. So different people will give different bending pattern. Different bending pattern means the position of the bands are different and the number of bands are also different. You can see that some of the specimen got less bands, like suspect 3 got less number of bands, whereas suspect 4 got more number of bands. So every individual got different number and location of the bands. So that gives us the unique genetic fingerprint. So why do they have got different number and location of the band? Is because there are different number and position of restriction sites. Remember just now we need to cut the DNA. So depending on how many number of restriction sites we have and the position of the restriction sites, and also different location and length of the VNTR or STRs. So later on, you know what does this mean? Because all these bands, right, are DNA which contain VNTR or STRs. So I'll explain this one afterwards. So this is another real case scenario. This is a sample from gel electrophoresis. So the bending pattern on the left side, so they call it DNA ladder. So this one, right, sometimes they call this bending pattern as the DNA ladder because it looks like that ladder, right? So usually the first sample is actually a standard DNA. So this is a standard DNA here, standard DNA of, that means DNA with fragments of known sizes. And then the rest of number 2, 3, 4, 5, 6, 7, 2, 3, 4, 5, and 7 are samples. What is number 6? Number 6 is again the standard DNA. So after you run the gel electrophoresis, then we can compare number 4. Where's my number 4? See number 4. Okay, number 7 is actually the sample from the crime scene. So this is sample from the semen, and semen has got 2 main types of cells: the sperm cells and the epithelial cells of the urethra. So the cells will contain the nucleus contain the DNA, so therefore we can run gel electrophoresis and get the bending pattern. So, and then this sample, so this one is DNA profile from defendant number 1. So you can see that the semen sample from the crime scene, right? Not from crime scene, actually from the victim. This semen is obtained from the victim that's been raped. Is matches. So this DNA profile matches with the DNA profile of number 4. So obviously this semen comes from defendant number 1. So you see, it's very powerful, isn't it? Now, another thing I want to bring to your attention is, can you see how come this band, right, is thinner, whereas these 2 bands are thicker? The location is the same, telling me that the length is the same, the length of the DNA fragment. These 2 are the same, these 2 are the same length. Why the thickness is different? Why do you think the thickness is different? Hello? Let's call Simba. Why do you think the thickness is different? So DNA fingerprinting, so this is to find out the possible father of a child. So this is mother, this is child, and this is father. So because the child's DNA comes from either the mother or father, so the bending pattern, meaning the genetic fingerprint of the child, right, must either match with the mother or else the father. So you can see that, so this one matches with the mother's one. This one, the mother doesn't have, this must come from father. So the father must have it. So this from mother, this from father. This one from mother, this one from father. Right? This one from mother, this one from father. So the child's genetic fingerprinting the band must match either with the mother or the father. So this is how we identify the father of the child. So these are other examples where samples where this is to find out the paternity of the child, means the father of the child. So let's look at the principle of genetic fingerprint. Now, genetic fingerprint, we use the probe which identify either VNTR region of the DNA or STR region of the DNA. So we want to know now what is VNTR, what is STR, okay? So GF stands for genetic fingerprinting, identifies VNTR stands for variable number tandem repeats, or is also called the mini satellites. Now, this is used in the past. So in the past, they identify the VNTR region for genetic fingerprinting. Now, nowadays, they identify STR for genetic fingerprinting. STR stands for short tandem repeats. It's also called microsatellite. So this is widely used now. So let us see what is the meaning of variable number tandem repeats. Now, variable number means different number of. Okay, it can be 10 to 20, different number of. So this is for STR. Tandem means clustered together and oriented in the same direction. Repeat means, for example, GT, when you repeat again, you got another GT, you repeat again, another GT, repeat again. So here you have got 1, 2, 3, 4, 4 repeats. Okay, so here 1, 2, 3, 4, 4 repeats for the base CA. So repeats, the repeats is made up of short base sequence of 2 to 5 base pair that will continue to repeat itself for 5 to 50 times. So what exactly is VNTR? So look at this diagram here. So this region here, oh, sorry, I clicked the wrong thing. Hang on. So where exactly is VNTR? So this section of the DNA is called the VNTR. Okay, VNTR. So what you can see here is VNTR is made up of variable number of, can you see you got this 1, 2, 3, 4, 5. So this one got 1, 2, 3, 4, 5, 6. So this one got 6 repeats, this one got 4, this one got 3 to 5 repeats. Repeats of what? Now this particular segment here is a length of DNA, which is between 2 to 5 base pairs that keep on repeating itself. So imagine if this is GT, so repeat again GT, 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 GT. So in this VNTR, it repeats 6 times. So this region is called the VNTR, variable number tandem repeats, because these repeated base sequences, they are arranged in a cluster, so it's called tandem, and in the same direction is GT, 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 GT. So this is VNTR. So in genetic fingerprinting, we use DNA probe that can locate the VNTR and bind to the VNTR region of the DNA fragments that have separated. So VNTR and STR are short length of DNA containing highly repetitive sequence of bases found in the non-coding region of DNA. So different individuals will have different genetic fingerprinting due to the differences in STR, because nowadays they use STR. So this STR can be found in all the 44 autosomes as well as the 2 sex chromosomes of the human. So different individual will have got different number of STR and different length of the STR. And then when we prepare the DNA for gel electrophoresis, we have to cut it into fragments. So now the chromosome also will have got different number of restriction site and different location of restriction site. All these will lead to the differences in the bending pattern in genetic fingerprinting. So therefore, different individual has different genetic fingerprint due to they have got different number and length of STR and different number and location of restriction site for all their 44 autosome and 2 sex chromosomes. So therefore, a genetic fingerprinting is basically used to reveal the differences in the number and the length of STR of the individuals. So therefore, different people will give different number and position of the bands. That is, they will give different distribution pattern, bending pattern, or genetic fingerprinting. So the Federal Bureau of Intelligence, the FBI in USA, uses 20 of this STR in its DNA database. That means they have got 20 different type of STR. So they actually use these 20 different type of STR to get DNA fingerprinting. So now let's put everything together so we try to understand what it means. So let's say, for example, this is the DNA, the chromosome from individual A, and let's say the chromosome from individual B. So you got two different chromosomes, and the red color band, the red color region here denotes the VNTR region or the STR region. Okay, so you can see these two chromosomes are different because of the number of VNTR. So I've made the length of the VNTR the same, only the number of the VNTR are different between these two. So I cut the DNA with restriction enzyme. So we assume that restriction enzyme will cut near to the VNTR region. So after I cut and run gel electrophoresis, what will I expect to get in my result? So I take this as an example, okay? So for A, I will get 3 bands, right? So let's say, for example, I run gel electrophoresis. Let's say I've got band number 1, band number 2, band number 3 here, okay? This is number 4, 5, 6, 7, 8. So I've got 8 DNA fragments of different sizes. For sample A, I put in this well, sample B, I put in this well here. Then I start the electric field to separate the DNA fragments. So for DNA from individual A, you can see that there are 3 fragments. So they will then separate into 3 bands, right? So number 2 is the shortest, so you move to the furthest away. Okay, and then number 1 is second shorter, so it will be here. And number 3 is the longest, so probably it moved very slowly, so it's here. So there are 3 bands in the gel. And then for individual B, you've got a lot of bands. So the shortest one is number 5. So number 5 is exactly same as number 2. So it's located in the same location here. So this is number 5. Then followed by maybe number 7, right? Number 7. So number 7 is shorter than number 1. So it will be moving faster than number 1. So it's here. Okay, then followed by number 4 is also shorter than number, it's the same. So number 4 will be here. Okay, then we have number 6, slightly longer than number 4, but very near. Number 8, maybe here. So these are the DNA fragments that are separated into bands. Then we use DNA probe to only bind to those DNA fragments containing the VNTR region. So that means to say it will only bind to this fragment number, this one here. And it will bind to this fragment as well, fragment number 5 and fragment number 7, this one here. So therefore, as a result, after you put the probe, you will only see these 3 bands. You will not see other bands because the probe only binds to these 3 bands. So now do you see that for chromosome A and B, they got different bending pattern because of different number of VNTR. Okay, in the same way, I want you to try to write the answer. So this is your homework. You draw out where all the fragments will be separated. So like just now, I number the fragments. So you draw the DNA bending and you number it 1, 2, 3 based on which fragment is it. And then you make it thicker, or you make it, or you highlight the band that the probe will bind. That means the band which contain the VNTR. So that will give you your genetic fingerprint. So you got a homework to do for this one. The next one here. So this is to show different length will give you different bending pattern, different length of VNTR. This one is to illustrate different number of restriction sites. So this one is exactly the same DNA, right? But they have got different restriction site. Sorry, they're not the same DNA. Huh? They got the same length of VNTR, but different number of restriction site. The bending pattern will also be different because of different number of restriction site. And the last example is to illustrate different location of restriction site. So again, these two DNA are exactly the same. They also got the same length of VNTR and same number of VNTR. The only difference is the location of restriction site is different. It will also give you different bending pattern. So this will be your homework for you to draw on the gel. So now we have got the father and mother to show a pair of homologous chromosomes, and we run gel electrophoresis. And after we run gel electrophoresis, you will start to see the bending pattern is different. So father has got 3 VNTR, so you got 3 bands. Mother have got 4 VNTR, you got 4 bands, and the location are different because the length of the VNTR, each VNTR is different. This is control. Control means what? Control could be standard DNA. Okay, so standard DNA, when you run the gel electrophoresis, if the bands are formed, that means your procedure has been done correctly. Just like when you do a COVID test, you must have got a band at least there to show that you've done the procedure correctly. If there's no band, that means the procedure is wrong, you can't see anything. So the control also helps us to see whether we have done the electrophoresis process correctly. Other than to help us to know, okay, what is the length of the DNA in this band? It's the same as the length of the DNA here. So inheritance matching, so we know that genetic fingerprinting can be used to find out who is the father of the child. So the reason is because for every individual, right, 50% of a chromosome is from the mother, 50% is from the father, right? Because 50% of the chromosome is from the egg, from the mother, 50% is from the sperm, from the father. So a child's VNTR may come from maternal chromosome or paternal chromosome. So the bending pattern must always match with either the father or the mother. So a child's genetic fingerprint will have some bands that coincide with the mother's band and the rest coincides with the father's band. So question, explain why siblings of the same parent have different genetic fingerprint. Okay, so let's say, for example, father and mother cross. Not all the sperm will get all the chromosome, right? All the sperms are genetically variable. So let's say this is you. So your chromosome from father is this one and chromosome from mother is this one. And then your sibling will receive chromosome of father this one and mother this one. So that's why your sibling can be very different from you, right? Because the sperms are genetically variable and the eggs are genetically variable. So you receive different combination of chromosomes. So all siblings are genetically variable. So your genetic fingerprint will not be the same. That's the reason, but they'll be very close to each other, very similar to each other. So the more the similar is the bending pattern, the more closely related are the individuals. Okay, so that's all for today's lesson.